Good afternoon, students. Welcome in Pharmacognosy practical. Today I will start the experiment number 3 and experiment title is to study microchemical reagents. Okay. In previous practical we have seen the experiment number 2, that is to understand the method of section cutting technique, staining, mounting and observation of section under the microscope. Now I will start the experiment number 3, that is to study the microchemical reagents. In that, prior concept is morphology and sensory characters of the crude drugs. New concept, position first, microchemical reagents. In that, microchemical reagents are used in the pharmacognosy practical for diagnostic identification of crude drugs, which stain the different components of TS of the crude drug. Okay. Then learning objective, intellectual skill. First is to acquire skill of staining and second is the ability to interpret from observation. Then requirement of this experiment is apparatus, microscope, watch glass, camel hair brush, glass slides, cover slip, beaker, dropper, filter paper, then forceps, test tube holder, tripod stand, wire gauge, dissecting needles, sharp razor, etc. Then chemicals of these experiments are microchemical reagents and water. Then crude drugs are root, stem, then seed, leaf, bark, fruit, rhizome, etc. to see the understanding of the procedure of staining. Okay. Then now I will start the stepwise procedure. In that, first is the study of microchemical reagents and second is the specific microchemical reagents. Now see, the first is study of microchemical reagents. In that, pharmacognosy includes the study of crude drugs obtained from natural origin. These drugs can be studied by their morphological or sensory characteristics and microscopical characters. Okay. For microscopical characters, they are treated with various reagents such as first cleaning reagent, then dehydrating reagents, bleaching reagents and mounting reagents. In cleaning reagents, these reagents are used to make the tissue clear in appearance. For example, chloroform, acetic acid, chlorhydrate and water. Cleaning reagents are used to make the tissue clear in appearance. For example, chloroform, acetic acid and chlorhydrate and water. Okay, these are the cleaning reagents. Then dehydrating reagents. These reagents are used to remove the water from the section or tissue and to make the tissue clear in appearance. For example, absolute alcohol. Dehydrating reagents are used to remove the water from the section or tissue and to make the tissue clear in appearance. For example, absolute alcohol. These are the dehydrating reagents. Then next is the bleaching reagents. Bleaching reagents are used to bleach the tissue or section and to make the tissue clear in appearance. For example, hydrogen peroxide is the example of bleaching reagents. Bleaching reagent is used to bleach the tissue or section and to make the tissue clear in appearance. Okay. Then next is the mounting reagents. These reagents are used to mount the tissue or section and to prevent the drying of sections. An example of mounting reagents are glycerin or mixture of the glycerin and water. Okay. Then next is the specific microchemical reagents. In that, individual staining solutions. First, name of the reagents, then components and observation. First reagent is eosin and component is cellulose and observation is red or pink color. Then next is the concentrated sulfuric acid with iodine and color components is color. Then observation are pale blue color. Then next is the fluoroglycinol with concentrated HCl and component is the lignin. Then observation is red or pink color. Then reagent safranin and components are lignin and observation is red or pink color. Then ruthenium red and components is the mucilage and observation is red or pink color. Then alcoholic picric acid, then component protein and observation is yellow color. Then next reagents is Sudhan red 3 and components is fixed oil and fats. Then observation yellowish brown color. Then next is the name of reagents is the dilute iodine and components are starch. Then observation is deep or pale blue. Then next reagent is concentrated sulfuric acid. Then components is saponin or stone cells and observation is green color. Then next is the name of reagent is ferric chloride and component is tannins and observation is blue or black color. Okay, these are the specific microchemical reagents, individual staining solution. First eosin, component cellulose, observation red or pink color. Then concentrated sulfuric acid with iodine, component color and observation is red, pale blue color. Then fluoroglycerin with concentrated HCl. Then component lignin and observation is red or pink color. Then safranin reagent, component lignin and observation red or pink color. Then ruthenium red and component is mucilage and observation is red or pink color. Then next reagent is alcohol picric acid, component is protein and observation is yellow color. Then next reagent is Sudan red 3, that is component is fixed oil and fats and observation is yellowish brown color. Then name of reagent is dilute iodine and component is starch and observation is deep or pale blue. Then next reagent is concentrated sulfuric acid, component saponin or stone cells and observation is green color. Then ferric chloride, component tannins and observation blue or black color. Okay, these are the specific microchemical reagents. Then next is the procedure. First issue the glass watches and add microchemical reagents to it. Then take the section of crude drug and transfer section in watch glass containing water. Then place section into watch glass containing microchemical reagents. Then keep it for specified periods and mount the slides and observe it. Okay, these are the procedure. Firstly, use the glassware, then microchemical reagents to it. Then take section of crude drug and transfer section into watch glass containing water. Then place section into watch glass containing microchemical reagents. Then keep it for specified periods. Then mount the slides and observe it. Okay, these are the procedure of this experiment. Then last observation. And here are write down your observation. Then conclusion. And last is the question. These questions are related to this experiment. Okay. After watching this video, you all students are answer this question. Okay, now I will stop here. Thank you. In next lecture, I will take the experiment number 4, that is to study morphological and microscopical characteristics of cinchona bark. Okay.